Neuroscience and beyond. Know more, get inspired. Welcome to the Neuroscience and Beyond podcast. My name is Willem and I am a PhD student at the University Medical Center Göttingen in Germany. Together with my PhD colleagues Sabina and Christina, we desire to bring science and especially neuroscience closer to the general public. Today, our guest is Constantin Pape. He is a junior professor at the University of Göttingen. Leading a pioneering research group, his primary focus lies in the groundbreaking development of deep learning-based methods for advanced image analysis. Among the topics that I am excited to learn more about during our conversation are the usage of artificial intelligence in image analysis of biological samples. Also, what are the challenges during such analysis and how we can use cutting-edge computer vision models released from Meta in image processing. To make our conversation even more exciting, we will discuss these topics in the light of neuroscience and microscopy. Before we begin, I'd like to mention that this podcast is separate from my research position at the University Medical Center Göttingen. This podcast is supported by the International Max Planck Research School for Neurosciences, as well as the Cluster of Excellence Multiscale Bioimaging in Göttingen. And now, please enjoy the talk with Professor Constantin Pape. Hello, Professor Pape. Thank you for being here.
Thank you for the invitation.
I'm looking forward for our discussion since I'm also involved in microscopy and I'm curious to learn more about the image analysis, segmentation, etc. But before we delve into the science, could you please tell us a couple of words about you and how did you end up in image analysis? We know that you're a physicist by training, but you switched to neuroscience, life science. What changed?
Yeah, I mean, I started with physics, kind of being interested in theoretical physics, right? Sort of really the string theory aspects of it. And at some point sat in a lecture there and was really overwhelmed by kind of both the math, but also the removal from actual reality, right? And this was kind of the narrative, but right, over some time I just realized that, this was not for me. And I was always interested in neuroscience quite a bit. I actually thought about studying neuroscience, but didn't really find anything that fitted. And then physics was kind of my other big interest, and I went for it. But then during studying, I heard a lot of computer science, right? Because just for whatever you do nowadays, right, computer science is pretty helpful. And after realizing that I was not really made for theoretical physics, I had no talent at all for experimental physics, it was pretty clear that I would go for something more computer science related. And then biology and neuroscience was really the application that interested me the most. And that's why I then started to work on connectomics, right? So I started with this in my master's thesis, right? We had a lab that was working on machine learning with applications in image analysis, and this EM connectomics, and I'm sure we'll speak more later about what this actually is, but I mean, just in a nutshell, right, it's sort of reconstructing the neural wiring diagram from big imaging data. And so this was really a application that fascinated me. And then, kind of while working on this, I really taught myself to program well, right? I mean, I knew this all during studying, but sort of, I was pretty overwhelmed in the beginning with it, but then during, 6 years of master and PhD, at some point, I kind of figured this out. I switched the applications a bit. I'm sure we'll talk about this more. I quite traveled around a bit. I was first in the US for a year, and then when I came back to Germany, I moved to IMPRS, which is still in Heidelberg, and is a big research institute, but was different from the university where I started out. And there I really came into contact with a lot of different biological problems, and a lot of them related to image analysis, right? And that's how my work kind of took a turn, first from physics, then to this pretty specific connectomics problem, and then going to EMBL to a pretty big spectrum of problems.
Mm-hmm. EMBL is the European Molecular Biology Lab, right?
Exactly.
It's a very prestigious institution. But you mentioned that you were also in the US. In which institute?
I spent a year at Janelia Research Campus, right? So that's also pretty well-known institute in neuroscience, located close to Washington, D.C. Mm-hmm. And this was really the institute that pioneered this EM connectomics, right? So meaning reconstructing the neural wiring diagrams, and now it's kind of lice, so of very small animals, from electron microscopy, so from very high resolution microscopy images, right? And the goal there is to understand, right, sort of the intricate connections in neural tissue at a much higher degree than is possible than with other techniques. And this institute really pioneered this, right? And so the lab where I started my PhD in Heidelberg at the university had a collaboration, right, to solve the related image analysis problems. And as part of the collaboration, I then went to this research institute and spent there a year in my PhD.
And now you kind of ended up in Göttingen. How did it happen?
Yeah, I mean, it was two interesting coincidences. So first, I am from Göttingen, right? So I'm from here. I moved to Heidelberg for studying, and I mean, with some interruptions, stayed till the end of the PhD. And then, at some point, like mid-PhD, I decided that I would give academia a shot, right? I mean, my work, due to, some good coincidences, let's say, ended up being fairly influential in a couple of areas, right? Sort of pioneering this large-scale image analysis for a few applications. So I knew that I had at least a chance to try academia. I wanted to stay in Germany, or at least in Europe, but then Germany is kind of the biggest attractor for science in continental Europe. And then when I finished my PhD, this position opened up. And it really, the focus of it matched my profile pretty perfectly, right? Because the focus was a junior group, or the position was a junior group focused on image analysis for microscopy. So more or less exactly what I had been doing and wanted to do. And then I just applied pretty much, I guess 3-4 weeks after defending my PhD. And this all worked out then to get the position. Yeah. I mean, maybe to say this is kind of, what I did, or a lot of my work is machine learning, deep learning related, which is kind of a very, large growing field right now. So this sort of transition from PhD to PI it's very uncommon in biology. In this field, is not so out of the norm, just because there's a lot of draw from industry, right? And so that means if people who stay in academia have a pretty good chance to start a group fairly early, because a lot of really good people also go to industry.
So basically there is a league of specialists, and you decided, let's give it a try, and therefore there is a high demand of specialists like you, and but that's great. It turned out to be perfect. You came to your city, where
back
you were
to
born.
my hometown.
So very nice. Could you maybe make a brief comparison between the working environment in Janelia, for example, and this one here in Göttingen?
Yeah, I mean, Janelia is a very intense place, right? I think that's the one-sentence summary, which means it's a bit, it's secluded, which sounds weird, right? It's kind of all around it is civilization, but it's these American suburbs where it's really hard to get anywhere without a car. So the campus itself is, kind of, if you don't have a car, you're pretty stuck there.
It's in Virginia, right?
Exactly, it's Northern Virginia, And also there are a lot of internationals, so there's then a very tight-knit group, right, of internationals who live on campus and who then, kind of mix work and private life, which kind of leads to this very, maybe unhealthy but interesting mix, and situations where people just go for dinner there and then go back to the lab is super common, which may in hindsight be not such a great idea for a long time, but for a few years I think a lot of people have a really good time because they can work a lot, make a lot of progress on projects, but still have like a very tight-knit group of friends there on campus, right? And I think this is kind of one reason why it is intense place, but also a place where it's easy to meet people with sort of the same interests.
It sounds some kind of inspiring, right? You have excellent scientists from all around the world, and all of them, they want to find something, discover or develop some technique, and you kind of work together. And for even if it's that intense for 1, 2, 3 years, then you have this network for the whole life, right? So you can establish collaborations for the future. So maybe it's desirable for people, especially from Europe, to go, because here the working culture is somehow different, at least that I hear and
no,
see.
it is very different also, right? Because it's very small labs. It's very collaborative, and I mean, as you said, this really leads to this very good networking. And I mean, of course, there are also people who are there longer and who also have a bit more of a established base there, and then have a different rhythm. But a lot of people are there for more like 2 to 5 years, and then really have this fairly intense focus stint there.
Do you know if they offer some like internships, some summer schools or something like that?
Yeah, they have pretty explicit programs for this. So they have some summer schools, and they also have student internships. So I think these are 6 to 12 weeks or so, and then for undergrads. Right. And so there's, I think, about 30 undergrad students or so each year that come for very high level, I mean, very sort of demanding, but also rewarding internships that will go there.
So to all undergrads that are listening, please check Janelia for summer schools and workshops. All right, so great. And now you're here in Göttingen, and you started recently your group as a PI. Could you please share with us what is your research focus?
And the research focus is building image analysis methods for microscopy data. So this means to automate tasks for extracting knowledge from imaging data, right? and particularly from microscopy data. I mean, there's a lot of different applications, but some example would be sort of having this light microscopes that image cells, for example, to image them while they are growing, maybe being treated with different drugs in different concentrations and so on, right, to understand the effect that some drug has on cell culture. Right, this could be used, for example, for testing drugs or for sort of checking how well some patient samples react to drugs. Right, and this can be done now with modern microscope with very high throughput. So this means, right, in parallel we have a lot of different experimental conditions, like different drugs applied to these cell cultures. And then if the sort of biologists or doctors that actually want to extract knowledge from this, that want to know how do these drugs affect these cells, would do this all manually, basically by looking at these images and somehow grading them. This would take a lot of time and could also introduce human bias, right? Or need even more time if they actually have to manually do measurements. And so the goal of my work is to automate such tasks, right? So for example, in this example I gave, to quantify how fast do these cells grow, or are they growing at all, or do they die, right? So this is sort of the underlying goal of my research, right? To enable automating such tasks, right? And this was one example, which has also some clinical applications. There are also a lot of more research-focused applications. We have, for example, working with electron microscopy, where you can see samples with a much smaller resolution and look sort of at very small parts of a cell, right? Like the cell nucleus, right, which holds the DNA. You can, for example, look at this in a lot of detail. And then the goal there is to really reconstruct this intricate detail to understand the processes that go on in cells to a much higher level.
Can we? So we dropped a lot of terms.
Sure, let's unpack this.
We can go into more detail, but kind of explanatory for the people who do not have experience in microscopy. For example, we have these light microscopes and electron microscopes. Maybe can we make a distinction between both?
Yeah, right. I mean, light microscopes are closer to the microscope that you would imagine if you think of one. So it basically means you have a sample, right, like some cell culture, and look through it with a microscope, right? So with optical lenses to sort of magnify the sample underneath, right? Maybe there's one important distinction there, right? There's kind of the normal microscopes where you just use normal light, and there's then also fluorescence microscopy, where actually you shine on the sample with a laser, and then there's like some small dyes, right? Like some coloring that these lasers sort of excite, and so we measure then the sort of emission of light from the cells that is excited by the laser. I'm mentioning this, right, because with this we can look at sort of specific parts in a cell, because these like small little dice, right? They can sort of be attached to different parts of the cell, right? For example, for DNA, one could then image only the DNA in the cell or only the sort of cell surface and so on, right? So this is kind of the light microscopy, where either we have like the kind of only looking with normal light, where we see kind of indistinct pictures of cells, or this fluorescence microscopy, where you have more targeted parts of cells. And all of this has sort of a resolution boundary. So this means, right, with light, we cannot look at structures that are smaller than a few hundred nanometers.
Can we put in the perspective the nanometer?
This is what I wanted to say. And like a cell, I mean, you're biologists, you know better, but it's maybe like let's say 10 micrometer, right?
Depending on the
cell, right? There's a huge range, but just as a order of magnitude, like 10 micrometer, right? And so if we would say we have 100 nanometer, that would be, okay, now I need to do math in my head, but would it be a thousandth of that? I think
100,
hundreds, thousands, right? But so basically, it means we can see this cell only up to some limited resolution, right? And this is the big sort of limitation in light microscopy. Of course, there's this huge caveat that there are also techniques to improve this in light microscopy. To keep it simple, I will not talk about them much more. I'm just saying what I say is right only true up to a degree. But now, if we go to electron microscopy, we use a very different technique to make these images, right? So instead of using light to look at the cells, we use electrons, right? So electrons are like this basic parts of atoms, right? And if we have a strong current, you can sort of get like these streams of electrons, and you can shoot with them at your sample, right? Again, right? The sample could be cells, and this can give you a much higher resolution, right? I mean, the reason for this is kind of physics. I don't want to go into this too much more, just to say we can get a much higher resolution, right, which is great, but it's not like we can look at living things with this. So basically we need some like contrast agent to make these images, and in this case contrast agents are heavy metals, right? So these electrons, they interact very much with heavy metals, right, like osmium or lead or so on. So in order to produce these images, what needs to be done is to infuse these heavy metals into the sample, and then they can be imaged, right? So this means if we have, for example, right, some tissue we want to image at a very high resolution, it can be done with electron microscopy by sort of fixing these samples, then introducing this heavy metal staining in there, and then using this electron microscopy beams to look at this, right? And so we cannot look at anything that's alive with this, which is possible with light microscopy, but we can look at much smaller resolution, right? So we said before, right, that we can get something like around 100 nanometers, just very roughly with light microscopy, right? With this, we can go down to maybe a nanometer, right? So we can look at structures that are a hundredth of the size.
Extremely small,
right? We can
imagine a different world, basically.
We can look at like individual vesicles in neurons or in nervous system, right? Where these are these tiny parts that mitigate the information exchange, right? And these are, I mean, the brain is extremely complex, so this means all these parts in there are very intricate, right? These vesicles maybe have a diameter of, I don't know, 40 nanometers, right? So this is tiny, but with electron microscopy, we can actually resolve this.
So to sum it up briefly, we have two different types: electron microscope, light microscope. With light microscope, we use light and fluorescence to detect some signal from targets that we already labeled with dyes or antibodies that were fluorescently conjugated as well. And in this case, we can use this microscopy to study living cells, for example.
Exactly. Yeah.
And in electron microscopy, we have electrons instead of light, and the huge advantage there is that we can go to a resolution up to nanometer range, which will give us details about things that sometimes we cannot access with light microscopy. And there are, of course, many other details, and when we say light microscopes in fluorescence microscopy, there are different types of microscopes, some of which we are going to hear in the next episodes with our further guests, but today the topic is a little bit different. So now when we have the microscopes, I'm curious to hear about what aspect. So when we say artificial intelligence, right, this is a hot topic right now. Everybody's talking about AI and computer vision and imaging, etc. But what aspect of artificial intelligence do you use in your daily work, for example?
Yeah, so what we use, I mean, if we need to give a name to it, right, it's deep learning. But so what we use it for is actually to automatically understand parts of these images. I mean, maybe I just start with an example, right? We just talked about electron microscopy and that we can see these vesicles, right? And so how this looks in the image, right, that we would have a lot of small round structures we see in these images. And these are these vesicles, right, that hold neurotransmitters. What exactly they do would go too far right now, but basically what is the question then is sort of how many of the vesicles are there, for example, and how large are they, right? And now if we would not automate this task, basically the biologist interested in there would need to go there and would need to kind of manually count all these vesicles. I mean, this would usually mean that you would have some computer program, of course, but then kind of put a point for each of the vesicles to keep track which were counted, and then sort of count these points, right? And for like a small scale study, this is fine. But usually this is done, right, to compare different conditions, right? For example, these samples, they're taken from animals after they die, right? And they want to study sort of different effects of some treatment, or it could also be just some tissue, right? And so to really make scientific discoveries based on this, we need quantitative data, right? So we need to count these vesicles in many different samples, right? And then this would be very time consuming to do all by hand, right? So what the artificial intelligence that we're developing should do in this case, right, is to automatically find all these vesicles so that we can count them and that we can also quantify their size, right? So that we know, right, that this treatment, for example, induces this change in the number of vesicles, right? And this can be used then to think about hypotheses, right, of what the actual mechanism is of some treatment, and how this affects sort of the neural information processing.
So you, in principle, someone gives you a lot of data, huge amount of data, and then you start building a model, training this kind of, okay, we're not there yet, but there's neural networks, for example, which learns to recognize some pattern and then gives you some output. Okay, in this particular image, we have this outcome, and this is that one.
Exactly, right? And this already brings us to one really important point here that I haven't mentioned yet, right? That, what we use are these, what we call artificial neural networks. This is because they are very, very loosely inspired by real neural networks. But it's basically kind of these, general structures that can learn to, that stay with images, right, to like map an input image, right, which again could be these images of vesicles to some output, right? So an example, the output could be the count of vesicles. And the important point here is that, right, we are not really describing any rules for how to do this, right? I mean, you could in principle think, right, one could go there and describe these vesicles. How do they look in terms of actual pixels and so on? If you actually try to write down rules for this, it turns out to be very hard and for any more complex structure pretty much impossible. So what these neural networks, or these artificial neural networks now do instead is, or what we do instead with them is to train them from examples, right? So basically we take pairs of the images and the correct count of vesicles, and then we train a network to predict this, right? And basically during this training, the sort of structure of this network gets updated in a way that it outputs the correct count, right? And I mean, maybe we can go into the details later, but the reason that they're called artificial neural network is that on a very abstract level, what happens during this training is similar to biological learning, right? I mean, if you go into the details, there's a lot of differences, but sort of as a first idea, you can think about it, this network, it predicts some number of vesicles in there, and we know the correct number, right? There will be some error in this prediction, and then we use sort of this, we tell the network that this prediction is wrong, and based on the sort of difference and the error, we can update the parts of the network to get closer to the true answer. This is kind of in a nutshell, the idea behind this. And this is machine learning approach, or deep learning, is what's underlying sort of a lot of the advances in artificial intelligence right now, right, from self-driving cars to ChatGPT on a very high level, right? They all implement some of this approach we just described. But in our case, right, we use these things to look at biological data, right, to automatically derive this analysis, like the vesicle counts we talked about.
And we're talking about these vesicles that usually sit in the synapse, in the presynaptic part of neurons, and they carry these neurotransmitters, as you mentioned, and due to neuronal activity, they release their content, and eventually there is a signal propagation to the other cells. And you're saying that these vesicles are around, etc. But when we look at for example, electron microscopy image, there is sometimes not that clear what is this and what is that, and it's very fundamental for image analysis to also segment the shapes and the forms.
This is the actual task that we are solving. Where segmentation basically means that we want to find all the different objects in our image, or the different objects of one type. Where again, in our case, right, this could be the vesicles, but it could, of course, also be other parts of cells. But this is the actual task that we would solve in most cases, right? So basically, what this neural network then actually does, right, it gives us the position and the shapes of all the vesicles or whatever we are interested in the image, right? And this we can then use to either just count the number that we have, or to really look at the shapes in more detail, right? To understand how some treatment induces changes in shapes.
Exactly, in the morphology.
Exactly.
Okay, but when we imagine now an image, this is a two-dimensional shape, right? So, but as we know, biological samples and cells, they are three-dimensional. And for example, electron microscopy, as also light microscopy, allows us to obtain the so-called Z-stacks, which is basically a volume, and we can look into different, it's called focal planes, and reconstruct the whole shape of a cell or neuron. And you already dropped this term, a connectome, which is actually exactly this. There is a chunk of a brain that is cut very thin and then combined, and people are looking into this data. And this data is extremely challenging to reconstruct, but you have been involved in similar projects, so maybe you can tell us more detail about it.
Yeah, exactly right. What you already said is in if we want to reconstruct this connectome, right? What we want to know is kind of how all the neurons in the brain are connected to each other, right? And I mean, they connect via synapses. I mean, we already discussed synapses in a lot of detail, or in a lot of zooming in via the vesicles and so on, right? But on a high level, the synapses just means, right, these two neurons, they talk to each other. They can exchange information via electrical impulses, right? And the synapses are very small, right? We talked already, right, about these length scales, right? Again, like a couple of 10 or maybe maximally 100 nanometer, right? So tiny. But this whole tissue that we would need to look at to reconstruct this now actual biological neural network is much, much larger, right? It can be on the order of centimeters. I mean, if we would talk about a human, even larger, but that's way beyond the current capabilities, right? So I think right now people are trying to go towards the mouse. But what is reconstructed already is the fly, right? And for the fly, this is, I don't really know, right? But the whole tissue is in the millimeter range, right? But so, and we need to resolve down to a level of few nanometer to really make out all the details, right? So you see this is like a difference of 6 orders of magnitude there, right? So this means this data is just giant that needs to be imaged and then needs to be analyzed. So this is one of the major challenges there. And it's also very multiscale, right? On the one end, we have these tiny synapses and we need to detect them, right, to really know the connections. And we have neurons which are these sort of very branched structures that go through large parts of this tissue, right? Because they transport information, right, from some part to another, right? To have some, I don't know, sensory input, right, from some, let's call it just nose, so some olfactory input, and then filtered to some higher point in the brain, right, where this is then processed, this input, right? So this is going through a large part of this volume, right? And we need to really reconstruct this neuron through this whole part. Then we need to find all the synapses on it and do the same for all the other neurons, right? And this is just a very complex task. So Davi Bock, who I work with in Janelia, always said, right, it's like a tangled ball of spaghetti, and you need to like follow all the spaghetti through this and find how they connect to each other. Yeah, so this is just a very challenging task on many levels because of the size of the data, but also just because the intricate structures that really need to be resolved in there. And then in the end with the goal to reconstruct this full diagram of how the neurons are connected to each other. And then this, of course, the hope is that with this we can understand brain function to a much higher level.
I think that this segmentation was done manually so far and not that much automated. And we have the fly and we have also the worm brain, right? The C. elegans. And I've heard about studies that they couple the function and then immediately take out the brain and cut it and look at the structure, and then you have a nice correlation between the functioning brain and also the structure behind it. But I wanted to ask you something related to the connectome. So we know that the brain is plastic, meaning that the synapses, these connections between the cells, they change on an even minute scale, hour scale. What is our, let's say, benefit if we know the full connectome of one fly in that particular moment, for example?
Okay, so first, I'll answer this. And right, just to say, I'm not a neuroscientist, but this is my perspective on it. Right, the goal would be to have a reference, right? So to have this map, right, this very, very detailed map for one fly, and then to map this to this functional data, right? So what you just described, right? These experimental recordings. And if we can actually understand, right, this activity that we are seeing now live, it corresponds to this part of the brain, right? Then we can actually combine this data, right? We can combine, okay, this activity we see live is happening here, and now we know roughly where this happens in the brain, and from the connectome we can actually derive, right, how exactly are these parts combined to other parts, right? And I think how well this works in practice depends a bit of how similar or different brains are between species or between individuals of one species, right? I think in some simple organism like flies, they're probably fairly similar, these brains. If we have like a more complex mammal, this might not be true anymore, but like to some degree, all of these share some same structure, right? And we know this structure from the connectome, and then the hope is by combining this with these other data from functional sources or also from genetic sources, that we can just have a more complete picture of how the brain works. I would just mention one thing, right? So you're right. For the worm, it was done manually, right? Because the worm so C. elegans, it has only 308 neurons. And this is super stereotypical. All of these worms have, I mean, it depends on male versus female, but I don't know which gender or which sex, but one has 308 exactly. For the fly, it wasn't done manually, right? They have like 50,000 neurons. Subparts can be done manual, but not the whole thing. And the whole thing was just sort of published, some version that was done automatically to some degree, and then proofread, right? Because this is such a difficult task. The automation is not good enough yet to fully do it, but it can sort of do most of it. And then the rest humans basically need to go there and correct it.
If I'm not mistaken, so to put this into perspective, let's say one movie, if you want to watch, is like 2-3 gigabytes information, right? And here we are talking not about terabytes, but petabytes, right?
So
of
for the
information.
fruit fly brain, it's 100 terabytes.
Terabytes, okay.
Yeah. But anything larger, you're very easily in the petabyte
Yeah.
range. So the biggest volumes they're looking in now, right, are parts of the cortex of a mouse, right? And this is in a few petabyte range. And I think the whole mouse brain would already be exabytes,
Oh,
right?
this is,
so
I think,
yeah.
still not manageable.
Exactly.
task.
This is still not manageable. But this is sort of right where the community is kind of hopeful with the advances in like the next few years that this will actually become.
Yeah, I remember Sebastian Sung. He's one of the pioneers of this.
Yeah.
He has very nice TED Talks about the connectome and mouse
Exactly.
connectome.
Yeah. He also has a very nice book about it, right? That's targeted toward the general audience.
So if you guys want to learn more about it, please go ahead and read it. So, but okay, there is this deep learning methods and imaging tools, but what would be maybe the biggest challenge for you in this regard, for example?
Yeah, so I mean, what we talked a lot about the neuroscience applications, right? I'm right now, a lot of what we're doing is actually more focused on these cell applications, right? Where we have light microscopy and looking at parts of a cell in there. And the big challenge there is that data comes from a lot of different sources, right? So basically, we already discussed this fluorescence, right? We can look at a lot of different parts of cells with the fluorescence, right? By sticking these dyes to different parts. There's a lot of different types of microscopes, as we already discussed, right? And so this means the appearance of this data changes a lot depending on the type of experiment. And as we said, right, these deep neural networks now, the artificial neural networks, they are trained on actual data, right? So they get like examples of the microscopy data and the output that the network should produce. And then this network, it will be specific for this type of data, right? So in the extreme case, right, we could say we have something that's trained on light microscopy and we try to apply it to electron microscopy. This can never work, right? But also in light microscopy, we have a lot of different settings that are somewhat similar to each other, but not similar enough that if you just naively train these neural networks, deep neural networks, on some sample, that they work for another sample from a different kind of microscopy. And there's so many different domains that, this is a big challenge, right? Because in principle, for each new kind of microscope, you would need to produce this ground truth data again. So this is what we call this training data. And this would mean in practice, right, we would tell the biologists we work together that, look, before we can help you, you need to spend a week and annotate all these cells for us, right? And you can imagine that if you actually want to spare the time, this is not great to come there and say, well, we can help you, but before that, you actually need to do all this work for us, right? So I think this is kind of the biggest challenge. And it's especially in light microscopy, where it has a lot of different applications, from clinical tasks to basic research tasks, a big challenge that we have just with like these kind of standard methods that I described. We really just learn how to segment cells or structures in cells for the given sort of type of microscope we train on. And this is really a big challenge, but also what my lab is focused on right now. So basically to develop methods that work more general, right? So that basically learn some more abstracted representations on cells so that they can find this in different types of light microscopy without being trained explicitly on this type. Right? This is kind of one of the big challenges and big focuses right now.
And then let's say you find a solution to this problem, then you develop a new software or a plugin to an existing software. How does it work so that the community can gain out of your...
Yes, this would be the goal, right? To really make this publicly available by having, there's these different kind of bigger tools that people are used, and then we would write these plugins for these, where the goal would be that the biologists using this, right, they would just need to mark like 2 or 3 cells, I mean, that's the vision, and then this neural network, which has like a general concept of what a cell is, learns from these just very few examples what the biologist is looking for in his or her sample, and this can then be applied, right, to all the experimental data that needs to be analyzed for a given experiment.
So now when you say this, it might sound very easy for the people who have no experience. For example, we segment shapes and size, but at the end you get data from, first of all, there are different microscopes. Every microscope has a different camera, different imaging modalities, different quality of the data, different formatting of the data.
Yeah. And different also organisms, right?
Exactly.
We could have human cell types or bacteria, right, which have vastly different shapes and vastly different sizes. Yeah.
We're talking about complex things that need to be analyzed. And biologists struggle. I'm a biologist. I struggle. I have to write my own code to analyze this data, then another code to analyze that data, and it's really challenging. And also there are still people who count manually a lot of cells, a lot of shapes, a lot of events, which is, of course, now with the existing technology, I think we can perform better, right?
Yeah, and that's the goal, right? To really make this technology that is there available to a much broader audience, right, of scientists who need these tasks. On the one hand, just by making them easier to use, and by solving this issue we just discussed, right, that these techniques, if you just train them on one type of data, will not generalize to new data.
Exactly. It's really fascinating, and also these tools are super helpful. As a experimentalist, I can say, and also the people behind camera, they work a lot with images, and it's we're looking forward to such nice developments, right? So you mentioned also during our talk ChatGPT. This is a different machine learning or artificial intelligence type from the neural network, right? It's a language model?
Yeah, it's a language model, but it's still, we would still call it an artificial neural network.
Artificial neural network, okay.
The structure is a bit different to the ones that we mostly use, but on a high level, the idea behind this design is still fairly similar, right? That is this kind of network structure of these sort of functions that interact with each other, and while learning, right, just these kind of connections between these functions are updated. The big difference in ChatGPT mostly is that it's really, really big, much larger than any of these models that we would use. And this means there's a lot more data needed and also a lot more compute to train it. And I think the big insight there is how it can be trained from language data taken from the internet, right? So basically to train these, the big companies that do this, like Google, or in the case of ChatGPT, OpenAI, they have these really, really big text data sets. And then these models predict the next part of the text based on the previous part, right? So just as an example, right, they could take some Wikipedia article, they sort of process this a bit to present it to this model, but they only show the first half. And then the model is is trained to predict the second half. And for this, we don't need any human annotations, right? The humans kind of collectively, as humanity, did this job by putting a lot of text on the internet. And then, I mean, we could also talk about the implications of this, but for now, let's just stick with the practical way how it's done, right? These companies take all this data, and then through this massive data set, and this sort of prediction task of the next part of the text from the previous part, these models learn some very powerful text representation, right? And then probably a lot of listeners have at least seen or tried ChatGPT once, right? It learns these chatbot abilities. I mean, there's some more things going on afterwards to actually get it to be like this good chatbot, but the main task is really just this training on this massive text data set.
Can we use this tool for image analysis, for example?
Not directly, but there is some work going on for integrating this. So for in the case of ChatGPT, right, they actually have the GPT vision now, which can also do some light image analysis. So basically, you can upload an image there and then ask it to describe the image, or also to do these image analysis tasks like counting things that we discussed. But it's, I mean, for specific things, it's still quite a bit worse than these specific algorithms we would develop, right? I mean, I just played around with this a bit, but for example, these counting tasks, it does a decent job, but it's quite far from like the specialized algorithms. And what is actually sort of implemented for these part of the models we don't really know yet, right? Because these are now, there's a race to commercialize this. So like OpenAI and Google, they don't really share all the details anymore. And for the just pure text part, right, ChatGPT, we still know fairly well what's happening there. But for the vision part, it's a bit unclear how exactly this is working, right? I mean, I think people reverse engineer this all the time. So it seems like for the image analysis, it actually writes a program and then runs the program over the image. This is what seems to be happening. But it's kind of the trade secret of OpenAI at this point.
Very interesting. And also this other company, right? There are now these tech giants like Google and OpenAI now and Meta, formerly known as Facebook.
Yeah.
They also released a very interesting tool, which is called Segment Anything, right? And you have used it already.
Yes.
Maybe you can elaborate a little bit on this.
So the idea behind Segment Anything is maybe to build something that's a bit like ChatGPT, but for computer vision, right? So for understanding images, and in particular to build a model that can like segment any kind of object in some image. And we talked about this problem already, right? That if these methods are trained for one task, right, like segmenting cells in light microscopy, but also segmenting cars in the real world, right, they will more or less only work for this task and for this kind of data. And the goal that the team from Meta had is to build a model that is more general, right? So that basically you can give it an image and either tell it just to segment all the objects in there, right? Which means basically find all the objects and their shape in there. And this is, of course, a very ambiguous task because what an object is, especially in the real world, but also in some microscopy data, is very ill-defined before giving sort of any more hints of what you mean by objects, right? It could be that each leaf of grass is an individual object, or that you just want to segment the patch of grass, right? But so they kind of came up with this way to train this model in a way that it solves this general segmentation task fairly well, and that it can also be guided by the user. So basically, as a user, I can just put a point, and the model will segment the object. So give me the outline of the object that matches this point best, right? So this is what this Segment Anything model is. And the way they did it is by building this giant data set where actually all kinds of objects are segmented in. It's mostly still actual images, but also some scientific data. And then they train the model for this. And what we then did, we got really excited by this because this could be a solution towards this problem that I just described, right? That we have all kinds of microscopy images and we need to retrain every time for it. But if we have a more general model that can solve segmentation in all these microscopy images, we would get much closer to the goal that we actually have. Right? And that's why we started working with this Segment Anything model and basically improved it for microscopy data. So this is kind of the important part there. The Meta, they published everything, or almost everything there, right? So unlike ChatGPT, where you can just interact with it, either through like this interface or through some programming interface, but you don't have access to the actual model, right? Meta just put this out there free to use and to improve upon for everyone. And we then basically implemented some new training procedure that improve it for microscopy data, right? And this is now kind of our step towards this vision, right, of having this one model where you can give it a few examples and it segments it, right? So right now, the main thing it can do, right, is that what I just described, you put some point and it segments the corresponding object, or it can also do this automatic segmentation without any points, but the real strength is this, right? With like a few clicks, you can put together very complex objects, right? And doing this kind of manually, right? Basically painting everything would take minutes per object. And with this, it usually works in 1 or 2 seconds, right? So that's kind of the main thing that, I mean, first of all, Meta achieved, and then by our work to improve it, we really made it a lot better for microscopy data.
Is there any plugin now that we can try?
Yes.
Okay.
There is a Napari plugin, right? So Napari is this Python tool for image analysis, and it's integrated in there, and we're still actively working on making this more user-friendly and also improving it. Yeah.
You mentioned Python, that is one of the programming languages, right? If you want to start now image analysis processing, would you recommend for people to start with Python and keep with it, or go to C+ or Java?
No, if you want to really do it a bit sort of wire programming, Python is definitely the way to go. Right? So this is a programming language that all these deep learning libraries use, but also a lot of other things that do like general image processing, right? Like just applying filters or things like this are all available through Python and its libraries. And it's much easier to use compared to C++ and Java. Right? I mean, because if you have some IT background and want to build like a big application, then things like Java and C++ are better because they're faster and they also enforce more structure on your code. But if you just want to like solve these scientific tasks like image analysis, then Python is really a very good solution for this.
What do you think about MATLAB? Because this is...
I'm an open source person, right? I'm
use
like
a lot.
But yeah, of course, right? People should use also what they know and what they have to. But if you're just starting new, right? And if you have the freedom, don't use MATLAB, right? It's commercial. It's worse in most aspects than the open source solutions now. So really, I mean, one way to go is Python. If you don't like that, right, there's things like R or Julia, which are similar but have different strengths that are also very good options. But use something that is open source and actually freely available. Yeah.
Unfortunately,
you're stuck to MATLAB.
I was taught by my boss MATLAB, but I will learn. I'm learning also Python.
I mean,
so
like
it's not that different.
It's not that different, and it always depends, of course, on the context. Right? If you have to work with code that's in MATLAB, and sure, this is a good thing to do. But if you have the choice, right, then I would say go rather for Python, or if you want to be cutting edge for Julia.
And now you have ChatGPT, which can actually switch and write you the code from MATLAB to Julia or Python or whatever. But I don't know why, but somehow in the community, in neuroscience, in decoding of signals and electrophysiology especially, a lot of MATLAB is used instead of Python.
I think MATLAB just really managed to sort of get into a lot of engineering disciplines and really was the major language there for a lot of time. And I assume that this just sort of also came into neuroscience, where a lot of like physicists or other engineers just started working and coding and then took this over. But I think in the last 10 years or so, right, the trend is very clear and it's going toward Python and more general open source solutions.
Okay, so take home message, start with Python and continue with other new tools and don't go to MATLAB. Of course, it's also very expensive.
I mean, this is the main reason, right? I think we have a lot of constraints, but as scientists, we should try to have things as open and reproducible as possible, right? And for like, I mean, at the German university, it's fine. You can get a MATLAB license, but if there's some, people want to reproduce it without the institutional background, right? This is already a pretty big barrier because these licenses are really expensive.
That is absolutely true, and I completely support your opinion on this topic.
But I mean, still also to say, right, it's not like I'm judging anyone using MATLAB. I said, if you have to use it, it's totally fine. But if you have the choice, I would always say, do something open source.
Of course. We know that, and we struggle with both. A little bit off topic, but I mean, it's actually into the topic. What do you think would be the next big advantage in the artificial intelligence field? We had like this neural network, which in principle are not new at all. They're in the 50s, Marvin Minsky and people like this, but now it kicked in a lot, and so this neural networks, deep learning, then we hear more about the language models like ChatGPT, large language models. In your opinion, where is this field going towards?
One really big trend is multimodality, which means combining text and images, and also in scientific domains, right? For example, combining imaging data on genetic data, right? To have these models that actually see like a more complete vision or more complete picture of the world by really combining different aspects of it, right? And I think text and language is really the prime example there. I already talked about this, right, GPT that now has vision. Another example, there are these image generative models, right? Things like Midjourney or DALL-E or Stable Diffusion. I think a lot of people have at least seen some of these images, right, where you can describe some image that you want to generate, and then these models generate some version of it, which usually looks good, might not actually match what you put in at all, but at least gives like some fairly photorealistic images, right? And this also really, this quality of these things improved a lot over the last 2 or 3 years based on these multimodal models, right? Which have like this joint, kind of representation of text and of images.
Okay, so I see. So they combine different aspects towards something more complex, and we don't even know what would be the next. The next step there really, for all the big players, seems to be this much tighter integration of text and vision. But also, I think in science, this has big applications, right? So having sort of structure representations and image data, or genetic representation and image data. And how exactly we do it, we still need to figure out, because this is fairly different than text and image in the details. But I think overall, right, this is where I see a lot of potential, because all these things we observe, right, the images in microscopy, the genetic expressions and so on, they are connected to each other, right? It's all just different views of the biological process. How do you think, as a physicist trained in neuroscience and with a solid background in computation and programming, how far are we from the generative AI? General AI.
I still think we are pretty far, right? These things will become more powerful, but they still lack agency. And I think it's still very unclear how to put in agency and also other things, right? Sort of like a real world model where you can sort of check your own predictions, right? Your ChatGPT outputs something, but it doesn't really have a concept of if this is true and false, right? And it has some simulacrum of this where people can ask it to reflect on these languages, on these answers, but people have probed it quite a lot and show that it doesn't really have a good understanding of what it says is actually true and false. And I think because these are just kind of giant statistical engines in the end, right? That see all of this data and learn all of these connections in them and then compress it down so that they have like kind of like a very low resolution picture of the internet or so in there, and can retrieve the information from there to answer questions or solve a lot of tasks. And this is super impressive, right? But I think it's fairly different from general purpose intelligence, what we understand there in humans or primates, right? Where you have really goal-driven behavior, right? And it's, of course, I think, fundamentally possible to do this algorithmically, but we don't really know yet. And I think these large language models, they will be a part of it, or these large multimodal models, but we still lack a few key ingredients, right? How to really put in, agent-driven behavior and this kind of metacognition that you actually know if your outputs are true or false and can reason about this.
Yeah, this even still we don't understand in humans how
exactly,
this agency works.
We don't understand it in humans. We don't know how to put it into computers.
Yeah. It will be. It's very interesting that some people they very bravely and a little bit stubborn, they defend this. We know we're gonna put it together sometimes, and others are like, nah, I don't think so. I think, right, it's physically possible. I'm a materialist, right? I think it works in the biological substrate. Fundamentally, it must work in the computational substrate, but biology is just so complex that we don't really know how to replicate it, and there will be a lot of breakthrough needed, right? So I'm fairly pessimistic that it will happen in my lifetime, and I hope there's still some way to go there. So who knows? Maybe the next model will help us learn.
And it's hard to predict, right? Because it could just be that it's like 2 or 3 more key insights, and we're there. But with science, these things are impossible to predict.
You never know. You never know. Yeah, science is fascinating, right? And that's why we're doing it. But now we can go to the more negative part of the talk eventually, where we talk about the problems in our community. So usually I start by dropping some of the problems and like reproducibility and people who leave academia, or the reviewing of papers, getting grants, etc. And so far we had the perspective of people who are heavily trained in life science, in biology or biochemistry. There was some also other people, but we did not have a person who is involved in computation. And I know that in this field things are a little bit different. And I would like to hear now from your perspective. You have already solid experience. What do you think are the problems in your field, for example?
I mean, one really big problem is just this growth in it, right? So if you look at the papers coming out in machine learning, deep learning, right, for a while they really grew exponentially. I think now we are at a linear pace, but it's still pretty fast. And this just makes it very hard to stay on top of literature, but also means that people repeat a lot of work, and also that people get pretty sloppy, right? Because, I mean, it's just impossible to have a full overview of the literature. And also, I mean, as in every field, right, there is this rush to publish and this pressure to publish. But coupled with this growth and this high output, this makes this even worse in a bit, because people don't have the time, right, to work on some paper for 3 years, as in biology. But if you have a new idea, you should try to get it out in 6 months, because chances are, if it's a rather obvious idea, 3 or 4 other groups are working on it. And this is not quite as bad in my niche, right, because not many people are working on microscopy images. But if you're having like general ideas in computer vision, machine learning, text processing, there's an incredible competition in there. And I mean, this has advantages, that it drives progress, but also has big disadvantages, right? That it encourages people to sort of take shortcuts. And I mean, I think most of the people are honest, right? Or most of the scientists there. But just this sort of external pressure just means that you're kind of forced to design your experiments and present your results in a way that is kind of more favorable than you would present them if you do it in a neutral way. I mean, this is kind of true everywhere, but I think it's just, a bit more extreme in the field. And just, this really influx of papers also leads to very bad reviews, for example. Right? So, I mean, one difference to life sciences is that in machine learning, most of the publishing happens via conferences. Right? So basically, there's these deadlines for conferences, and then the actual papers are submitted there. And there's thousands of papers submitted, right? So for the big ones, it's like easily 10,000 papers. And then you need, of course, still 3 reviewers per paper. But the field has grown so quickly that there are not enough experienced people to stem this reviewing load, or they are super busy. So you get a lot of low quality reviews, right? Of either people who just pass through papers because they don't have the time, or that barely read it and then don't understand it and kick it out due to this. And this just makes this whole process very noisy, right? And I think this means we have kind of similar problems than in the life sciences. They're just a bit different because the pace is a bit faster in there.
Okay. I did not know that people are actually submitting during conferences.
Yeah, most people are surprised because it's fairly different from most other sciences. But these are really the major venues for publishing. And some of these are now, if you would like look at these measures like impact or so on, fairly up high in there, right? Because there's so much being published in these fields.
And then when you submit it to a certain conference, does your paper go to some journal, or this is some kind coupled to the?
They have proceedings, right? Conference proceedings, and these are more or less like our journals, right? So basically, for Nature, for someone working in deep learning, would be NeurIPS or CVPR, right? So these are like just names of big conferences, and these are the super prestigious places to publish, and they're conferences that then bundle all the papers that get accepted into proceedings there.
That's very interesting. Yeah. And I guess there's also a fairly high fee to submit your paper, or
submission is for free, but then to actually get it accepted, you need to go to the conference, and the conferences have fairly high fees. But this is actually still a lot better, right? So like this maybe like 800 euros or so on as a conference fee, which is compared to 10,000 for Nature, nothing, right? So this is actually one big advantage that, it's still fairly expensive, but you also get out much more, right? You actually go to the conference and you have the full conference experience and get your paper published for this.
Does it make sense? So life scientists, they publish, also medical scientists, they publish their paper before they're peerly reviewed on a website called bioRxiv, so that
we have the original of this, which is archive.
Archive, right? Okay.
Right, so this comes from physics, right, and was the first big preprint server, right, where exactly what you described, people would publish their things before a peer review, and now archive has been adopted by the machine learning community as well, right? So I mean, I don't really know the numbers, but I would estimate around 60 to 70% of the papers get published first to archive.
Okay, and these other papers or journals that you mentioned that compares to Nature for biologists, for example, are they open source?
Yes. Yes.
Okay, this is a big difference. It's a big difference because in our field, for example, we first need to do the research, the big work, submit, eventually get it published, we pay the fee, and then this is not open source, meaning that other people and institutions need to pay to this company so that they get access to the research, which was funded by the taxpayers. Very interesting model.
I mean, like I said, there are also a lot of problems in this community, but a couple of things that are really positive. That is this, right? Basically, all research is open source. So these conferences, right? They all have some way to make it available. I mean, it's usually it is published by some of these big names like Springer, but they have some special deal that they can host it somewhere else, and it's the same version of the paper. And this is sort of the whole spirit of the community is fairly open, right? So also this sharing code, even sharing code before publications and so on, is still quite common, right? I mean, it's also not each paper and everywhere, but it's still all the good papers in the end will share code, because otherwise people will not trust it, right? So if you make a claim that I have the best method and have these benchmarks in your paper and no one sees the code and no one can reproduce it, people will just dismiss papers. Because, like I said, people just show their experiments in the best light. And so to really have an independent evaluation, you need to be able to reproduce this. And in that sense, there are reproducibility problems, but the fact that things are shared and people actually try to reproduce, and this is fairly easy because it's code and it's not dependent on any experimental condition, this makes it possible to really have independent reviews of papers. And of course, this doesn't happen for the whole mass of papers, but for the big ones that claim to be a big achievement, there's often replication efforts in days after things being published.
And what do you think could be a nice solution to the problem with the this pace of publishing and this pressure to publish and to get funding maybe?
I mean, it's a tough question, right? So I mean, specifically to our field, right, I think it will plateau at some point, because I mean, infinite growth is not sustainable. And I think we are a few years from this, and then it will plateau and I think get better, because also, right, people will or the scientists will get more senior, there will be more reviewers and so on. I think the more general question is, of course, how can we have a more, in a way, equitable system where funding isn't tied to just publishing, which is both very stressful for an individual researcher, but also kind of detrimental for putting out good science. And I mean, I don't really know, because this is kind of the metric we use right now. And it's really hard to replace it fully by something else. I'm afraid I would say I see the problem, but I also can't really give a good answer, right? I mean, there's things like eLife, which has these experiments of doing publishing differently, right? That you basically the paper goes out and then it gets reviewed, but this is more collaborative, and it really, the review process happens in the open. And I think all these ideas are good, but it's very hard to see how, right, on a large scale this can affect and change the system.
You are fairly early in your career as a PI, but do you think that you could encounter a problem where you have a project and you want to hire someone, but due to the competitive salary outside of academia, it's hard for you to attract people?
Oh yeah, I mean this. Everyone has this problem all the time. So basically, it's still okay on the PhD levels to find PhD students, but finding postdocs in our field is really, really tough. So I mean, this is kind of I don't have that much funding yet, so I am have not looked for postdocs myself yet. But I think as soon as I will encounter this problem, and I think people just don't fill positions in the end. This happens fairly often due to this.
This is very sad, right?
Yeah.
Because it hinders you to advance in your research, and therefore the whole community suffers. It's unfortunately how it is, and we need more funding.
Yeah,
but,
exactly, right. But in this field, like I said, especially hard because there is so much interest from industry. Right, we cannot pay competitive salaries, and we cannot even offer better working conditions. Right, this is another big problem, of course. So there I think academia also needs to change a bit. Right, I mean, it's hard to argue on this with the salaries maybe on an individual level, because like you said, this all comes from taxpayer funding. But sort of making the whole job a bit more, positive with work-life balance is I think something we should really strive for. Also, this is hard to do just on an individual level, but at least there's a bit more, flexibility to not have unreasonable expectations on people.
Yeah, but I don't know. I mean, I think the general public should be more aware, and we try to do this with this podcast, right? To kind of convey science in a more simple way, and then convince people that this is important, right?
Absolutely.
And in most of the times, basic science, it's a long-term journey. It's not like you discover something tomorrow and you develop a drug tomorrow, but if you don't do it, then you cannot advance at all in the future. So yes, it's the outcome of our job is sometimes quite long-term, and even we might not see it in our lifetime. We never know, but if we don't do this research, then we will hinder the progress of our species, right? And if we convey this message to the public, right, then they know that we are funded by taxes, meaning the government need to allocate more funding for research, for example, then we can get more funding, and eventually these people can get paid more. But someone has to bring the awareness.
No, I think it's true. It's really important, right, for all of us as scientists to really communicate this, right? And like the deep learning field we talked about is really the best example. But these techniques, they are pretty old. They were invented maybe the start of this in the 50s, then kind of the groundwork of everything was laid in the 80s, but then the interest in this really stalled because we didn't have the data yet and the computational power to train this, right? So this was kept alive for 30 years only by academic, right? By researchers who were interested in this, right? Who thought these are good models for learning, to sort of model intelligence. And then, like I said, only like 10, 15 years ago, this field exploded because we were somehow, with the internet, with all the data that's out there and the computational resources, these models started to work, right? But if there was not kind of this basis of like really just maybe few dozen scientists who kept working on this, right, this would have never happened. And all of this happened without much industrial or commercial interest that would fund it before that.
And also, if we go to the physics, all the physics that
and this
has
is
been
true
discovered
everywhere in basic science.
Technology is basically physics, right? So it's incredible, and I think this is a nice positive way to finish our podcast episode. Thank you very much again for coming and for sharing with us your research and your enthusiasm. Also, it was a pleasure to talk to you, and I hope to see you around in Göttingen to conferences or cluster, the events of the cluster.
Yeah, I will join this one more, hopefully.
Thank you very much.
Thanks.
Once again.
And thanks a lot for the invitation.
Sure. Neuroscience and beyond. Know more. Get inspired.